Good afternoon, everyone. First of all, let me thank you all for giving me this opportunity today. My name is Chikara Mizukoshi from Nagoya University Hospital, Japan. Today, I would like to present single-cell survival analysis using a deep generative model. First, I'll explain the background of this research. Many methods exist to link gene expression with clinical outcomes. However, current approaches are limited to bulk or cell type resolution. And there are no methods that link clinical outcomes to each single cell. How cost of single-cell RNA sequence limits the ability of large-scale cohorts with clinical information and single-cell sequence data. Utilizing cohorts that have previously collected bulk RNA sequence data linked with clinical information could serve as a viable alternative. Next, let me talk about our method. We developed SC-Sub, a deep generative model for single-cell survival analysis. This method assigns a contribution to hazard for each cell based on bulk RNA sequencing data and survival time information from large cohorts. The bulk RNA sequencing data is decomposed to the single-cell level using single-cell RNA sequence data as a reference. Then, hazard function is regressed by the Cox proportional hazards model using the proportion of each cell in each bulk. SC-Sub performs deconvolution using single-cell RNA sequencing data that contains the same cell proportions as the bulk RNA sequencing data. Single-cell RNA sequence data from patients not included in the cohorts can be used as a reference. The single-cell RNA sequencing data does not need to be linked to survival time. This is an overview of SC-Sub model. SC-Sub has 3 steps. SC-Sub uses VAE to run the latent cell states. And then SC-Sub runs the proportion of each cell in each bulk and its contribution to the hazard based on the latent states. In the first step, SC-Sub reconstructs the single-cell RNA sequencing counts using the VAE framework. Through this process, we obtain low-dimensional latent cell states, which contains denoised key information about the cells. In the second step, SC-Sub decomposes the bulk RNA sequence data to the single-cell level using deep color framework. Deep color is our previous method to decompose spatial transcriptome data. The proportion of each cell in each bulk is estimated from the latent states. In the third step, SC-Sub estimates each cell's contribution to the hazard function. The contribution is estimated from the latent cell state. The optimization is performed using the same framework as Cox proportional hazards model. Here, I explain the Cox proportional hazards model. The Cox proportional hazards model is a multivariate survival analysis model. The hazard function indicates the likelihood of an event occurring at a particular time. No specific function is assumed for the baseline hazard function. We present the formulation and loss function in the third step. SC-Sub regresses hazard function using the Cox proportional hazards model. Here, the covariates are the proportion of each cell in each bulk. The regression coefficients can be interpreted as the contribution of each cell to the hazard function. Let's move on to the results. First, let me explain the actual data we used. Currently, the Cancer Genome Atlas TCGA provides gene expression, mutation, and epigenomic data for hundreds of patients across many cancers. These data are linked to clinical information. Our aim was to use this public data to perform an integrated analysis of clinical information and single-cell data. Next, we applied SC-Sub to 12 cancers. As a result, in 6 cancers, the prediction accuracy for test patients outperformed random estimation. For these 6 cancers, we proceeded with downstream analysis. We applied SC-Sub to the melanoma dataset. Using UMAP, these figures display cells in 2 dimensions. Each point represents a cell. Some cancer cells and stroma cells showed a large contribution to hazard, and this suggests that these cells have a negative impact on prognosis. We investigated the correlation between the contribution to hazard and the expression of each gene. This allowed us to extract genes that show a negative or a positive effect on prognosis. Then we performed gene ontology analysis using the top genes. The results show that the immune system has a positive effect on prognosis. This one. Consistent with previous studies, macrophages with high SPP1 expression showed a negative effect on prognosis. And macrophages with high CXCL9 expression showed a positive effect on prognosis. Next, we extended our model to spatial transcriptome data using renal cell carcinoma data. First, we trained SC-Sub to run contributions to hazards in renal cell carcinoma. Then we decomposed each Visium spot and assigned contribution to each cell. Finally, we assigned special hazards to each spot based on the proportion of each cell in each spot. This is the visualization of special hazards across histological section. This provides valuable spatial insights into tumor prognosis. Spots with higher proportion of cancer cells tended to have higher hazard score. This may be because cancer cells create a microenvironment that negatively impacts prognosis. The interactions between cancer cells and stromal cells in the tumor microenvironment are well known. We focused on T cells and used the spatial information to extract cancer cells co-localized with T cells and investigate the intercellular communication between them. This analysis allows us to identify key genes involved in cell-cell communication and investigate how these genes influence hazard. In conclusion, SC-Sub is a novel method that links single-cell RNA sequencing data with clinical outcomes. Using the TCGA dataset, SC-Sub can estimate the contribution of individual cells to hazard functions. Lastly, I would like to express my heartfelt gratitude to the staff of this conference. I would also like to extend my deep thanks to our lab members. Thank you for your attention.
Are there any questions from the audience? So to the best of my personal knowledge, it seems to be that you developed the first approach that incorporates single-cell information into the prognostic, like hazard ratio Cox regression model. Is this the first type of the model for predicting the patient survival, hazard patient survival?
Yes, yes.
At a single-cell level.
Yes, there are no method to link clinical outcomes with each single cell level. Yes.
Contribution, yeah, the quantitative contribution at single cell level. So, because in TCGA there are up to 33 different major type of cancer, I think pretty much you can like develop like a tissue specific or cancer type specific hazard ratio, regression model tailored for specific, cancer type, at the single cell level. But why, when you benchmarking your model, you only utilize like slightly more than 300 patients? Why? Because I think in TCGA there should be thousands of cancer patients with survival information, right? Yeah.
Yes. This model can't use the cancers which don't have many patients.
So have you also like try to incorporate other relevant clinical information, for example, in addition to the single cell, but there are also other different type of omic data as well as like pathological images.
Yes.
So pretty much you can integrate all this into your Cox regression model in order to further improve the prognostic prediction of cancer patient survival.
Yes, there are many extensions of our model. In the future, it would be interesting to map clinical outcomes on HE tissue images, or we can use multi-omics data to link clinical outcomes with omics.
So, any other questions? Yeah.
Thank you. It's a very interesting talk. I was just wondering how well the prognostic clusters that you identify map with like the cell type clustering, and if that information can be used to improve clustering.
So you mean we have to validate the spatial hazards?
So I was meaning how well these prognostic regions match to the original clustering and if this could improve the original clustering.
Sorry, I didn't cluster the special spots.
Any other questions? Yeah. Yes, please.
Thank you for your presentation. My question is that do you think single cell data or bulk data can be used to make clinical decisions in practice?
So, yes, our model allows us to data-driven discovery of cell populations that affect clinical outcomes. So we can extract cell populations and genes to affect clinical outcomes. So this model can be used as a feature extractor.
Okay, thanks.